Okay, take two of doing this video. I messed up the first time because I did not properly close my door, and then I proceeded to not download the file properly. So yeah, I have my water, so my throat doesn't kill me. So if you hear me taking a break, you know why. Alright, let's get into this then, yeah? So, we're going to talk about these. I already talked about these actually quite a lot, so I'm not going to go over them too much, but yeah. Yeah, stuff. Already talked about this, right, with the microRNAs. Feel free to send me questions if you have questions. My email's in the first video of the 440 series, so you can just send me emails or come to the IC, either one. Two ways to make the microRNAs for research. We've talked about this already as well. You can either order them from a company or use a plasmid. So, yep. Already talked to you about translational control. Talked about how the polio virus is this weird thing. Under normal conditions, you do this, but the polio virus has an enzyme that cleaves this, and so you end up with this for the polio mRNAs, stuff like that. Talked about this a little bit. Don't think it's too important to know this. Just know that there are some genes and mRNAs inside of your cells that naturally use internal ribosomal entry sequences. And then you can use them in also other techniques, like for trying to transfect a cell and stuff like that. If you need a little bit more context on that, feel free to take a look at my previous lecture video for that. So, lecture 3 is going to be covering protein trafficking. And yeah, now we're into this portion a little bit. So, let's go ahead and get into this. So, already talked about this slide a little bit. Already talked about this a little bit. We're going to mainly focus on this section, which is why it's highlighted. And I already talked about this. There's vesicular transport and protein translocation. So these are the two things to take into account. So, exocytosis, endocytosis. I hope by now you know what these are. As someone who's done neuroscience for 2-something years at this point, 3 years at this point, I am getting tired of seeing these. But basically, exocytosis is this process where you have a vesicle and it's released into the external environment. So cytosis is the process of release, if you will, or cytosis is the process of combining with a membrane. So exocytosis, external release of this thing. So this is relieving. Endocytosis is the opposite. Endocytosis is endo going into cytosis, the membrane, so into the membrane. So this right here, if you look at this, we've got a case of exocytosis right here, followed by a case of endocytosis right here. And I already talked about this before. Remember, from ER to the membrane, what's in the cytosol stays in the cytosol, right? Cytosol, cytosol. Talked about this a little bit. So we've got anterograde and retrograde transport. Anterograde, remember, is from the ER. So anterograde would be going, I wish they showed it better, but anterograde is going this way and retrograde is going this way. In this thing, they've labeled it as secretory and endocytotic, which should make sense, right? Secretory, secreting from the cell, it's going to leave the cell. Endocytotic, endocytosis, it's entering the cell, staying inside the cell. There's a whole bunch of stuff going on here. Jeez. Already talked about this. Things like clathrin, COP1, COP2, very important vesicle coating proteins. They help with initiation and stuff like that. Here is anterograde and retrograde stuff. So, if you look at this is a very confusing image. What I want you to remember, red arrow is anterograde, blue arrow is retrograde. So, the green arrows are actually also retrograde, which is weird. But basically, anterograde, 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 retrograde is all of this other stuff. Retrograde, 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 retrograde. So you can see here that for the most part, when it comes to retrograde transport, we're using a lot of COP1, so these blue dots. And we're also using some clathrin. So clathrin and COP1 are used in retrograde quite a lot. On the other hand, though, if you look at this, you can see that COP2 is used in anterograde transport quite a bit. But there are cases where COP1 is also used. So you can see here COP2 right here for retro or anterograde, and also there's a COP1 right here just doing anterograde. So it's a bit mixy and matchy. I wouldn't say that one particular one drives is like favors the one or the other, but if you want to think of it like specifically, you guys mainly focus on this region, so I wouldn't worry too much about this area. I think you should probably just understand that generally speaking, COP1 does anterograde and clathrin tends to do retrograde in this area here, but I think it's more important to focus on this section. So if you're going from the ER to the Golgi, COP2 is for anterograde and COP1 is for retrograde. I think that's the thing that you should definitely keep in mind. Okay, so we talked about clathrin a little bit before. I want to kind of reiterate the importance of clathrin. So with clathrin, what you should kind of keep in mind is kind of drives vesicle formation. And so I know I said up here that you often use it for retrograde transport based off this image. But like I said, it's kind of all over the place. So, I mean, even though I said it was retrograde, you can see retrograde here. But, I mean, it's doing anterograde right here. So, again, it's kind of wishy-washy for that. So I would say just think of clathrin as like a general purpose coat protein that helps with both retrograde and anterograde transport. And then you can think of COP1 and COP2 as like anterograde or retrograde if you want to think of it like that. But they're not really set in stone, if you will. So here we have, clathrin drives coat formation. So here's an example of how this looks. I'm not going to lie, don't really think it's that important to know this entire thing. I think what you should know for sure is, we have these receptors. These are cargo receptors. These receptors then bind a protein that they want, and then an adapter protein binds to that receptor. And then once that happens, these adapter proteins bind to clathrin, and then these clathrins keep binding to each other over and over again. They connect to each other in these portions here, and eventually it forms a full circle. And then these fission proteins bind here, and they cut this thing off, and then you end up with a fully formed vesicle that eventually releases all of its clathrin and stuff like the adapter proteins, and you just get this vesicle. Cool slide. I would actually probably write this down or memorize it. I don't think it's too hard to memorize. I mean, just remember, you have a cargo receptor protein that holds the protein that you're trying to transport. You bind the protein to it, bind the adapter protein, bind the clathrin to the adapter proteins, do it a bunch of times, cut the vesicle from the membrane, kick off all these adapter proteins and stuff like that. All these are out of there, end up with your vesicle. Okay, so here we've got monomeric GTPases control coat assembly. All this is really saying is that, the formation of these coats uses GTPases to do so. Here we've got an example of this. So we've got ARF GTPases for these and SAR. These are two different GTPases. Monomeric meaning one subunit, and they control coat assembly. So you can see here, COP1 and clathrin both use ARF. and COP2 uses SAR. So here's an example of, let's see here. This is SAR, actually. It's just examples from they don't actually show a picture of ARF, do they? ARF is here somewhere, I thought. Huh, okay, well, that's fine. But basically what's happening is, like, this right here is basically the mechanism for how we get these adapter proteins to attach, plus the clathrins and all this. So basically, these things trigger all of this stuff. That's the whole point of these. So, what to keep in mind from that? If you... realizing that I like doing them. Okay, so if you look at this, you have this inactive soluble SAR1, and you have this amphiphilic helix, all this jazz, yada, yada, yada. I think the main thing that I want to show here is this is like a Jeff, right? So this Jeff here, after this SAR1 has hydrolyzed its GDP, needs to replace its GTP with a GTP. So it does that. Now we have an active membrane-bound SAR. Now this SAR can be used alongside COP2 to form a vesicle. So, see here, we bind this. This is now active. Notice how we have this Sec24 and we have this SAR1. So now in its active form, we can form a complex with this cargo receptor, and then once this hydrolyzes, we can form a vesicle. That's kind of the main idea. Basically, all I want you to get from this is these guys, these GTPases, are what allow for the formation of vesicles. So notice here again, I'll reiterate it. We use a Jeff to switch GDP for GTP. We make this active SAR1. This active SAR1 is now what's being used to create a vesicle. So now this active SAR1 is here. We can hydrolyze the GTP. We can form a vesicle as a result of that. Okay, vesicular transport. Rab GTPases allow vesicles to target specific organelles. So, RABs. What are RABs? RABs are G proteins that are found on vesicles and direct them to the right place, like molecular zip codes. So, a RAB is like a zip code. Cool. Basically, it acts as like a flag. It tells the vesicle where to go. So, there's a whole bunch of different RABs. As this note says, you do not have to memorize them. They're pretty cool, though. So, the other thing that you should keep in mind about vesicles is snares. Snare proteins are incredible. They are so wacky, and oh gosh, I don't even want to think about them. But here, we have a couple things to look at, actually. So, the first thing I want to draw your attention to is actually this. So V snare and T snare, what are they? So V snares are, basically these snares are like traps. So basically when the vesicle is trying to fuse, it uses the snares to like pull itself and combine. So a V snare is a vesicle snare, and a T snare is a target snare. So a target snare is on the target for where the vesicle is trying to go. So if a vesicle is trying to fuse to a membrane, the T snares would be found on the membrane, and the V snares would be found on the vesicle. So I'll show you that in a second. Here is an example of, this is a nice image actually. This is a nice image showcasing the entirety of the fusion process, basically. After we've done this, it does not show this part. But once we are here, we have our cargo receptor, we have our cargo. What's happening is, remember, we have a RAB GTP. This RAB tells us where this vesicle is going to go. Pretty cool. So now we have a couple different processes. Remember these. There's tethering, docking, and fusion. Tethering, docking, and fusion. Jeez. Okay. So
How does this work? First, the tethering step. This is where the RAB is really important. What happens here is the RAB is like a flag, like I talked about before. There is a RAB effector protein on the target for whatever we're looking at here. If you see what's happening here, this RAB effector protein is binding to this RAB. And that basically initiates the docking process. In the docking process, what happens is the T snares and V snares start to twist around each other and collide. And that causes the RAB effector to bend. So now you have this. After that, we have the fusion step. From here, what happens is the RAB hydrolyzes its GTP into GDP, and it dissociates from the vesicle. And so it also binds to this GDI, which I can't remember what GDI actually stands for anymore. I think it's supposed to be something like guanosine dissociation inhibitor or something like that. I can't remember. But yeah, it binds to this GDI as a RAB GDP, and that allows for the fusion of the vesicle. That's it. If you were to copy one slide from this section about fusion of vesicles, it would be this slide. This slide covers everything. So, already talked about this. V snares are for vesicles and T snares are for targets. Not going to reiterate that. And here are some diseases that are actually caused by changes in vesicle fusion. Causes paralysis, muscles can't contract. Botulinum toxin actually basically cuts the snares and stops vesicles from being able to be released into your muscle, like the synaptic clefts, so you actually can't contract them. And then this is the opposite. Well, no, it's not the opposite. It's pretty much the same. It's this time, so this targets cholinergic, so it targets acetylcholine neurons. It means that they can't contract because what's happening is you snip the snares, you can't push acetylcholine into your synaptic cleft, it'll never bind to your muscle cells, they'll never contract. Tetanus is actually kind of the opposite. Tetanus causes muscle paralysis because it does the same thing where it blocks, where it cuts the snares, but it cuts it in the GABAergic neurons. GABAergic are inhibitory neurons, so basically they're supposed to inhibit muscle contractions, but what happens with tetanus is because you snip the snares for the GABAergic neurons, they never inhibit the muscle, and so the muscle just contracts continuously. This is just a cool little slide showing you what's going on here with the with how these things are working. So VAMP, syntaxin, and SNAP20. So VAMP is a V snare, syntaxin and SNAP25 are T snares. These are the targets of botulism toxin and tetanus. These are actually things I learned about in neuroscience, which is how I know about them. Here is an example of, with and without the toxin. So normally this is what happens. With the toxin, you cut these, right? And so see how you can't really, you're never sending any signals here, so you can never contract. So, believe it or not, this is how Botox works. Oh yeah, there's two types of cargo for vesicle stuff. There's selective and non-selective. So non-selective cargo is just anything nearby gets picked up, and selective cargo is just there's specific receptors in the vesicle that can only bind certain things. So that's all it is. So you can either have specific receptors that bind certain proteins, which is selective cargo, or you can just have a vesicle that just swallows everything around it. So here is actually a fun little vesicle trafficking example using LDL receptors, which you guys talked about in 441. So if we see this here, we have our LDL, our cholesterol, it's in our blood, and then this LDL binds to a cell. Not cell, binds to a receptor on our cell. What happens is, after enough of these LDLs have bound to these receptors, you get these clathrin coat proteins and adapter proteins that form here, and they bud to form a vesicle. And once the vesicle is formed, these things all kick themselves off, remember, like I said. And so what you end up with is this vesicle, which turns into an early endosome, a late endosome, and then a lysosome. So basically what I'm saying is this thing matures, this vesicle matures over time, and eventually the lysosome is an organelle where it degrades things. So the lysosome basically degrades the cholesterol and the vesicle so that it can be turned into free cholesterol to be used by the cell. And you can imagine that there are many different mutants for this. So, you can have a ton of different LDL mutants. You can have a case where, it's normal. Maybe you have a mutation in the receptor region. Maybe you have a mutation in the clathrin region, so it actually never endocytoses, right? So maybe it never comes into the cell. Like I said, so maybe it's normal. Maybe it doesn't have a binding region. Maybe it never endocytoses. Maybe you just have no receptor. Could be any of them. So now we're going to talk about signal sequences. So signal sequences can be a lot of different places. They can be at the end term, they can be inside the middle, they can be at the C term. And basically what's important about signal sequences is that they decide where the protein ends up, or the product. So maybe a certain signal results in the nucleus, maybe another results in the cytoplasm, maybe another results in outside the cell. Changes depending on the sequence. And so an application of signal sequence is targeting GFP to the nucleus. So yeah, basically what they did here is they took GFP, which is generally a cytoplasmic protein. They added a nuclear localization sequence, which, believe it or not, is a tag for the thing to go into the nucleus. And lo and behold, what they end up with is GFP in the nucleus. Shocking. So basically what they found is that there are certain tags that tell proteins where to go. So like I said before, we have our GFP, we add this NLS sequence, we end up with GFP in the nucleus. So they found that there are different tags for different places. This. Memorize. Just memorize it. Or write on your note card, whatever you want to do. This you must know. Basically, import into the nucleus equals a bunch of positive charges. Export from the nucleus is leucine isoleucine rich. Import into the ER, extremely hydrophobic. And then return to the ER is KDEL. Return to the ER is a bit of a weird one. I never really fully understood how this works. If I'm not mistaken, I think it's like going from like the Golgi and the membrane back to the ER, but it's a bit of a weird one. It's also usually a C-terminal sequence, if you notice that here. So now here, I want to reiterate this again. This is very important. Please, 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 please write these down. Okay? Just to make sure I hammer that into your heads. Write it down. Important to know. Okay, protein trafficking, like I talked about, so we already talked about this a little bit. So let's talk about the nucleus. How do proteins get into the nucleus? Remember that the nuclear envelope is double membrane, so it's kind of a weird structure. All sorts of random stuff to it. There's the cytosolic fibrils, there's the outer and inner nuclear membranes, there's nucleoporins, there's scaffold nucleoporins, a whole bunch of stuff. Basically, these are all very structural things. There's also the disordered region of the nucleoporins. So what you should very much keep in mind is the disordered region of the nucleoporins, so like these little hairs, and the cytosolic fibrils, or fibrils. These are very important. So, this is why it's important. When we are looking at protein trafficking into the nucleus, we're going to talk about trapping. So trapping, generally speaking, what happens is most large proteins, they hit the membrane of the nucleus and they bounce out. they bounce out. Very small ions, like sodium, potassium, these things, are able to pass directly through. However, proteins with a nuclear localization sequence get grabbed by these fibers, so like these fibrils here and these disordered hairs. And so basically it like tags it and pulls it into the nucleus. So I wish they showed it here, but they don't show it here, but there should be hairs here. Ions are small enough where they can avoid these hairs, but proteins are just large enough where they are able to get grabbed by these hairs if they have a nuclear localization sequence, because they're complementary or whatever you want to say. They form interactions. Similarly, these hairs also kind of work as a shield. So if there's no nuclear localization sequence, proteins will just hit them and bounce away. Like, right, similarly, ions that are super small pass straight through. And then, like I said before, if the protein has an NLS, it'll basically bind to these hairs, these fibrils, and it'll go through because it's recognized by the fibrils. And here we're going to talk about chaperones a little bit. I'm not going to harp on it too much. Basically, proteins, after they've been folded by the ribosome, after they've been released from the ribosome, they fold, right? So you have a peptide chain, it grows, you make a protein. Cool. And then with this ribosome, right, you take all that jazz, basically the HSPs, these are just protein folding proteins. They are proteins that help other proteins fold. So essentially what's happening is as the ribosome is coming out, these proteins are binding and they're helping the protein fold. So they have this ATP on them, they hydrolyze, and that hydrolysis is used to fold the protein. And so you do this over and over again, and eventually you get a correctly folded protein. Or the other option is you do all of this, you keep hydrolyzing ATP, you keep trying to fold it, and it doesn't work, and you get an incorrectly folded protein. If that's the case, there are two options. You can either go back and restart and try and refold it properly using the HSPs and ATPs, or you can use chaperone proteins like GrowES. GrowES, basically what it is a trash can. It's a trash can, and it tries to fix it. It's a trash can that tries to fix things. That'd be a cool invention. Basically, this chaperone, what it does is it takes incorrectly folded proteins, puts it in one side of it. This is a hydrophobic protein binding site. So it should make sense why it's hydrophobic, because remember, proteins have hydrophobic regions on the inside. And so the way that, GrowES is able to recognize incorrectly folded proteins is because incorrectly folded proteins will have hydrophobic regions on the outside. And so because there are hydrophobic regions on the outside, that actually allows this GrowES to bind to these incorrectly folded proteins. So incorrectly folded protein goes in here with all its hydrophobic regions exposed. The cap gets put on. This thing attaches some ATP. It uses the ATP hydrolyzes the ATP, and tries to fold it correctly. And then it releases the correctly folded protein. So, yeah. There's a couple different processes that happen here. So basically, when you make this new protein, usually it's correctly folded without help. If it needs help, it's correctly folded with the help of chaperones. If it's not correctly folded with the help of chaperones, what happens is it's incompletely folded. The chaperones tried its best. It just gets digested by the proteasome. And if all else fails, and somehow this protein is still able to live, even after going through all that, it ends up becoming a protein aggregate, and that's where you get diseases from. So actually, that's the entirety of this lecture. Again, I feel like I'm going quite quickly with these. I do apologize for that. I hope it's not too fast for you guys. But I'm trying to just make sure I get the most important points out here without going over the lectures like fully for another 50 minutes essentially. But yeah, again, if you have questions, feel free to put them in the comments. Come to the IC, ask me them while I'm still here. But yeah, other than that, good luck and happy studying.